and welcome to the podcast titled The Next Trip, Developing the Second Generation of Psychedelics and Their Analogs for Targeted Medical Use. In today's session, we will discuss the preclinical, clinical, and regulatory requirements and strategies that need to be considered for second-generation psychedelics in development for the targeted indications. The potential of use of psychedelics for the treatment of various CNS indications is currently under active investigation. Some researchers are already examining use of modified chemical structures and analogs for psychedelics in an effort to produce efficacy and mitigate potential side effects of these medications, which we refer to as the second generation of psychedelics. A formal drug development pathway is required when developing a drug for a targeted medical use. Given the nature of psychedelics and their promising therapeutic potential, various strategies can be implemented to explore the efficacy and safety of these novel drugs in both the preclinical and clinical setting. Today's podcast will examine several strategies that second-generation psychedelics may utilize to differentiate their pharmacological profile and strategically demonstrate efficacy and safety data that may differentiate from first-generation candidates. You'll be the first today to hear what the leading industry experts have to say on this rapidly evolving field. My name is Beatrice Setnik, and I'm the Chief Scientific Officer at Altasciences. I am joined today by a panel of well-known experts in the field of CNS drug development and research, and I will have each introduce themselves, starting with Dr. David Heal.
Hello, I'm David Heal. I'm one of the executive directors of DevelRX Limited. We are a CRO which provides consultancy support to the pharmaceutical industry, particularly in the development of CNS active drugs. My background is in neuroscience. I trained at Oxford University and subsequently joined the pharmaceutical industry in the mid-1980s. And I was responsible for teams that developed drugs in metabolic disorders and schizophrenia. After I left, I joined Renasci, which was a consultancy company we set up, and we were offering laboratory services in the CNS area for the next 18 years. And in that time, we managed to help clients get 10 additional drugs onto the market. Since 2020, I've been one of the directors of DevelRX, which is a company that I helped found.
Great, thank you. Next, we have Dr. Christopher Atterwill.
Hello, my name's Christopher Atterwill. I'm a pharmacologist, first of all, and then I became a neurotoxicologist working at Oxford University in the Department of Clinical Pharmacology. I have experience across experimental and applied neurotoxicology, and I've worked as a GSK Professor of Toxicology at a UK university, where I helped pioneer organotypic in vitro models for looking at in vitro neurotoxic effects. I've also been a Director of Drug Safety at blue chip UK CROs and pharma companies responsible for the preclinical development of new molecules. As an aside, I'm a practicing and registered UK pharmacist with experience in clinical pharmacology. Thank you.
Thank you. Next, we have Dr. Deborah Kelsch.
Hello, I'm Dr. Deborah Kelsch. I'm a board-certified psychiatrist and a principal investigator located in Kansas, currently working for Altasciences. I trained at the University of Kansas Medical Center in the Department of Psychiatry and was on faculty there for 13 years. It was during that time that I started working as a sub-investigator and then also part-time at Altasciences, which prior was Vincent Associates, doing primarily phase 3 trials in psychiatric conditions. In 2007, I started full-time working at Altasciences doing phase 1 and 2 trials. At that time, we had transitioned mostly to that. So I'm the principal investigator really for any trial that involves drugs that affect the central nervous system or special populations like opiate use disorder or recreational drug users. as would be required in a human abuse liability study. So thank you.
Thank you. And lastly, we have Dr. Denise Millivan.
Hello, I am Denise Millivan. I am a clinical psychologist, a neuropsychologist with 20 years of experience in working with complex neurological and psychiatric conditions. I am also a research scientist that has been in the field of phase 1 clinical trials, looking at the potential for abuse and the impact of novel molecules on behavior and cognition since 2007.
Great. Thank you so much. And thank you for joining today. This podcast today is brought to you in partnership with Altasciences, a contract research organization offering early phase drug development services and research, and DevelRX, a consulting team of experts in drug development supporting the pharmaceutical and biotech industry in the development of new drugs to treat psychiatric, neurological, and metabolic disorders. So let's get started with our first topic. First, we'll review the regulatory requirements for developing a CNS active drug and how those requirements specifically relate to the second generation psychedelics, taking into account abuse potential evaluation, the drug scheduling process, and some of the unique attributes that CNS drugs have in development. So perhaps let's get started with some of the preclinical considerations that a sponsor may need to take into account when developing the second generation psychedelics. And Chris, David, I'd love to hear your comments on that.
Well, one of the things which is very, very clear about the second generation psychedelics is they're going to be treated very much as any other CNS active drug. The FDA has made several discussion points about this at various meetings, saying they don't want to set up a unique set of barriers to the development of these particular compounds. That being said, they do pose certain specific challenges, and I'm sure we're going to get into those in greater detail later in the talk. But in terms of the regulatory package that you'll need to conduct for these molecules, it's the simple package that you would look at for any other safety pharmacology assessment. That means you're going to look at, in the case of drug abuse, you're going to have to conduct a drug discrimination study, looking at the discriminative signature of the compound. You're going to conduct an intravenous self-administration study, looking at the reinforcing potential, and you're also going to have to investigate whether the drug has got a potential to cause physical dependence on cessation of repeated dosing. Chris, what do you think about the other issues related to the safety of these drugs preclinically?
Yes, in the preclinical development of a new molecule of a second generation type, there are 2 or 3 specific toxicological considerations to take into account, and I'm going to cover those a little bit later with some slides. But specific areas, of course, are neurotoxicity, which was observed with the first generation molecules, cardiotoxicity, and then other potential toxicities on peripheral systems in the body, which may impact, of course, on what's known as the serotonin syndrome seen in the clinical setting. Now, those studies can be, to some degree, built into the preclinical early studies supporting progress to IND and phase 1 trials. And we'll talk a little bit as well about how we can make bespoke studies and investigative studies to cover those eventualities.
And you raise a really good important point, Chris, that's really related to the timing. So for the audience that may have less experience with drug development, can you describe the types of preclinical data that's generally needed for an IND versus the type of preclinical data related, for example, to abuse or dependence potential that may be collected throughout later on in the drug development process, even in parallel to some of the clinical studies that are conducted?
Of course, I mean, David is going to cover in more detail, I think, the abuse potential studies, what is known as the safety pharmacology package for this type of molecule. But in addition to that, to get to that stage, using a traditional preclinical development package, one would need the basic genotoxicity package for the molecule, that's in vivo and in vitro, range finding and acute toxicity studies, that's single dose, and also toxicokinetic studies to build onto those to look at the exposure potential of those compounds and the exposure plasma levels to relate to dose. And then, of course, one needs to conduct the 1 month or 28-day studies in 2 species. And I'll touch on that later because I think the choice of species is quite important in considering some of those points I mentioned about neurotoxicity and cardiotoxicity also.
Great, thank you. And then once an IND is filed, this enables the critical next step in the drug development process over to the clinical evaluation. And usually we would start with a second generation type of molecule in your first in human studies, where you're evaluating safety and pharmacokinetic exposure in humans, and then progressing to clinical studies in phase 1, 2, and 3. So throughout the clinical development program, you'll be doing standard types of studies to evaluate the bioavailability of a formulation, the pharmacokinetics, but you're also looking at very important elements. Of course, efficacy will be coming through later into the phase 2 studies where you have proof of concept and then larger patient trials that will look at specifically if the drug is effective in the target indication. But there are also many other important clinical questions that arise, and those are things related to, if there are perhaps cardiac effects and if there's any QT prolongation. There are dedicated studies that may need to be run. The regulators are certainly interested in any kind of drug-drug interactions, particularly cognizant of the type of patient population that the drug is targeted for and what that patient population may be exposed to in terms of concomitant medications. So certainly from that safety perspective, we're also interested in the human abuse potential studies, as we will get into later in this podcast. But those types of studies and the abuse potential and dependence potential are integral to the drug scheduling process, which is really the process by which data is reviewed, and ultimately agencies such as the FDA, in collaboration with other government agencies like the Drug Enforcement Agency, will determine based on data if a drug needs to be further controlled through the Controlled Substances Act. So this is all very important and applicable to psychedelics. So I will open it up to my colleagues. Dr. Millivan, Dr. Kelsch, a lot of cognition and some very important questions we ask early on in drug development around the characteristics and pharmacology of CNS active drugs, and I'll have you comment on some thoughts here.
Certainly, I think that if we are specifically targeting hallucinogen drugs and psychedelics in particular, one comment that immediately comes to mind is the psychedelic drugs have not traditionally been found to have so many physiological effects, kind of like when we are thinking of opioids and respiratory depression, or when we are thinking about stimulants and cardiac risks. Psychedelics have not necessarily been found to have so many physiological risks, but they have been found to have quite a significant psychological risk in the sense that they elicit an emotional reaction that might trigger some risky behaviors. And for that particular reason, I would think that looking at the possible impact on emotions and also on cognition, on the safety, for example, to drive later on after having taken a psychedelic molecule, that is something that one would have to keep in mind.
Thank you. And Dr. Kelsch, from a safety perspective, as you're investigating these types of drugs, particularly early clinical research, what are some of the medical safety considerations that you think through as you're evaluating these in the clinical setting?
Right. So anytime you do a phase 1 or 2 study, the subjects are generally in-house and we have lots of supervision. And I think it depends on whether the population is naive to recreational drugs or has experience If they're naive, you're looking for sort of events of special interest that might suggest that there's some sort of abuse potential, like somnolence or dizziness. Whereas if you have a experienced group, then they can they'll give you other terms. I feel high, I feel drunk, so you have to sort of tease out those sort of events, but it's just anticipating, understanding the potential side effects of the drugs and to be alert to those. And then if they have some reaction that's psychiatric in nature, to lots of reassurance and perhaps prolonging their in-house stay until that's resolved. And just good documentation.
When developing a second generation psychedelic, these are really, some novelty is introduced to the molecule, whether it's an analog of an existing psychedelic or it's a completely new drug entity. The FDA generally will treat these as new chemical entities, and therefore the onus will be to develop it and to investigate it like any other new chemical entity, as opposed to being able to supplement some of that information, as in the case with first generation psychedelics, where there's some published literature and already safety information that's characterized about these drugs. So that's, I think, one important distinction on the regulatory requirements that differ between some of the first generation, your psilocybins, your MDMA, LSD, where we have some body of literature to show the characteristics of these effects, which can be to an extent supplemented as part of the regulatory filing. But of course, it's very important to identify critical data gaps in order to be able to fulfill the regulatory requirements for a targeted medical intervention. So for our next topic, we will discuss the required preclinical and clinical studies that we need to consider for regulatory submissions, as well as the timing of their conduct. And more specifically, we'll be discussing the methodological considerations that we need to consider both for the preclinical and clinical studies when we're designing and conducting such studies, particularly as it relates to the new psychedelic drugs.
So thanks very much for the introduction, Beatrice. In this slide, what I've put up is a proposed screening strategy for the non-clinical evaluation of the abuse potential for novel psychedelics. Now, the process is very similar to that for existing CNS active drugs. When we think about the psychedelics, we really are thinking about two separate classes of drugs, each with their own individual challenges. So we have the entactogens. These are drugs like MDMA and their congeners. And we have the classic hallucinogens like psilocybin and the other 5-HT2A agonists. And the first real challenge when you're looking at these drugs is actually to determine whether they have hallucinogenic properties. Now, as I'm going to show you in a minute, we have a couple of ways in which we could do that. The first is using the head twitch behavioral model in rodents. And this is one of the best tests that we have in order to detect hallucinogenic properties. But also you can use techniques like drug discrimination in early stage testing in a non-GLP mode. And for example, you could use a 5-HT2A agonist cube drug discrimination using a selective agonist like DOI or DOM to detect whether the compounds that you are developing will actually generalize to that cube. And then we move into the GLP safety pharmacology testing, which includes the drug discrimination testing, looking at the psychoactive signature of the drug. For this, MDMA is an excellent cue for detecting all types of psychedelics. Then you can use, once again, in a GLP mode, 5-HT2A agonists, such as DOM or DOB, to look at hallucinogenics. We have the intravenous self-administration test, which looks at the reinforcing or rewarding potential of the drug. And here there are certain factors that need to be taken into account. the selection of the FR schedule, the difficulty that the rat has to offer in terms of work in order to get each drug infusion. You've got to think about things like the test duration. How many sessions are you going to allow for stable responding to occur? And finally, you've got to think about some refinements. And one of the refinements that we're going to talk about is the use of progressive ratios and breakpoints to determine the relative reinforcing effect of a drug.
And David, if I may ask, in terms of the reinforcing effects, do the psychedelic drugs behave differently in these models compared to some of the classic drugs with known abuse potential, like the opioids or the amphetamines? If you could comment on that.
Well, that's a very interesting question. I mean, in terms of reinforcing potential, you know, the entactogens like MDMA, and I'll show you some data later on, will perform exactly like, you know, conventional positive reinforcers. They are relatively weak, but they certainly will maintain self-administration in both rats and in primates. The hallucinogens are slightly different, and this is where we get into some of the more philosophical and strategic questions associated with these drugs, because traditionally it's always been assumed that the hallucinogens do not maintain self-administration in animals. However, you have to consider that the number of papers that have been published on this topic is relatively few, and the extent to which this topic has been investigated is not in any great depth. If, for example, you think about, well, the hallucinogens are not actually reinforcing, you then have to think about the predictive validity of that result when it comes to human subjects. And this raises the interesting philosophical question, because actually hallucinogens are not taken for their euphoriant properties. Human abusers actually enjoy the mind-expanding and mystical experience of hallucinogens, but that's very, very different from the euphoria which is actually elicited by either stimulants or opiates. And for this reason, actually that negative result that's been reported for the reinforcing effect of the hallucinogens may actually be the correct, valid translational prediction of their euphoriant abuse potential in humans. So let's look at the head twitch behavior. One of the things about this particular model is that it's often associated with 5-HT drugs. However, if you go back and look at the original literature, and here we're going back to a paper by Korn and Pickering back in 1967, what you can see is that the paper was entitled "A Possible Correlation Between Drug-Induced Hallucinations in Man and a Behavioral Response in Mice." And what you can see here on the left-hand side is that two classical 5-HT2A agonists, LSD and phencyclidine, both induce head twitch responses in mice. However, if you look at the panel on the right, what you can see is that a number of drugs like yohimbine, atropine, and hyoscine, these are drugs which are not 5-HT2A agonists, will actually elicit head twitch behavior in animals. And there is a substantial and significant correlation between their ability to produce head twitch behavior in mice and the dose which produces hallucinations in man. So this is a very important model when it comes to looking at hallucinogenic potential across drugs with a wide range of pharmacological actions.
And David, one question, are there other classes of drugs that also elicit the head twitching, or is this something that's unique to these hallucinogenic type properties in this particular model?
It seems to be specific to hallucinogenic properties. All of the drugs which I have ever come across when using this model over the years have been hallucinogens. But for this, it has to be said that it depends what you find depends on where you go looking. And we've not really conducted extensive evaluations of drugs which don't have hallucinogenic properties. What you can say is, things like opiates or stimulants don't produce this behavior in animals, and it certainly seems to be common to the hallucinogenic drugs. In terms of the serotonergic hallucinogens, I put up a selection here of drugs which have been shown to produce head twitches in mice or rats. And you can see that there is a very, very good level of correspondence between drugs which are hallucinogenic in humans and the eliciting of this behavior in animals. So for 5-HT2A agonists, this is a really good model in terms of looking whether the drugs are hallucinogenic or not. Now, when you move on to the safety pharmacology testing, you're then moving into the GLP testing arena. And here you're going to think about drug discrimination. And one of the things which is quite interesting is the use of MDMA as a discriminative cue in animals. What makes MDMA particularly interesting is that it will detect as generalizing to the drug discriminative cue, both the entactogens, as shown here on the left-hand side in blue, but also the hallucinogens as shown in green. So from the point of view of having a big safety net for looking at psychedelic drugs, this particular discriminative cue is actually very attractive because it means you can capture or detect the psychedelic properties of both subtypes of drug in this particular area. Now, one of the very, very important issues when dealing with intravenous self-administration is to think about how we are going to adapt this model to look at the reinforcing potential of drugs which are very different from the classical stimulants like cocaine and opiates like heroin. And one of the challenges about these drugs is you have to set the sensitivity of the model according to the reinforcing potential of the drug you're looking at. And historically, the FDA guidance has indicated that a fixed ratio of 10 for a response requirement, which is 10 lever presses by a rodent on a lever in order to get each drug infusion, is the appropriate response requirement to be used. But what you can see here is that when you are dealing with drugs that are weak reinforcers, and I've got MDMA in the middle here, actually this will only maintain reinforcement on very low schedules. And in this case, we trained the animals on heroin first. And you can see that MDMA is reinforcing across quite a range of doses. But this is on an FR3 schedule. It's not on FR10. And why do we have to do that? Well, the answer comes in the following slide. This shows the breakpoint for reinforcement. And on the left, we can see cocaine, classical Schedule II stimulant. And on the right, we've got MDMA. Now, the breakpoint is the maximum number of lever presses that an animal will make in order to get a single injection of the drug. And the more reinforcing the drug is, the bigger the number of lever presses. What you can see on the left is that cocaine is highly reinforcing, and it will take, at high dose, an animal will make between 100 and 150 lever presses in order to get each infusion. But if you look at MDMA on the right, you see a very different picture. Here you can see that the maximum number of lever presses that an animal will make in order to get a drug infusion is down in the 30s. And if you look at the panel on the right, you can see that all of the drugs which are moderate reinforcers and weak reinforcers have breakpoints of 30 or less. This means that an FR10 is far too onerous a schedule to use when you're looking at the reinforcing potential of these drugs. And for that reason, what will happen is you will generate false negatives. And this is not a situation which is acceptable when you're looking at a major safety signal like abuse potential of these compounds. That's the preclinical work. And I think now it's important to think how this is going to pan out in the clinical setting.
Thank you, David. And I wonder, before we move on to the clinical considerations and methods, if you might just briefly mention the physical dependency studies and what's known in that with respect to psychedelics and potentially second-generation psychedelics, and how those are assayed in the preclinical setting.
The physical dependence liability studies are really looking at the consequences of terminating repeat dosing. And it is applicable both in terms of the use of drugs in clinic. We know, for example, that opioids can produce physical dependence even when used at clinically effective dose. And we also know that it's very, very important in terms of dose escalation and physical dependence when these drugs are abused. In general terms, the psychedelics, whether they are the entactogens or the 5-HT2A agonist hallucinogens, do not produce the classical physical dependence liability signs that you see with drugs like opiates or benzodiazepines. However, this will have to be investigated. And the way we do this will be very similar to the methodology that we use for existing CNS drugs. So, for example, you need a positive control, and here it really doesn't matter whether it's an opiate like morphine or a benzodiazepine like diazepam. The role of that particular drug is simply to act as an internal validation for the method. What is really important is what the drug itself does. And for this purpose, you generally administer the drug at a plasma exposure level that's similar to the clinical Cmax, and a multiple thereof, usually 3 to 5 times, for a period of 28 days. Terminate dosing immediately, and then look at the physical, physiological, and behavioral signs of withdrawal, usually over a period of 7 days. And here you will be looking for key measures like weight loss, reductions in food and water intake, and signs of distress and loss of condition in the animals. So in this regard, if you've got a reasonably comprehensive set of measures that you're looking at, there is no reason to treat these drugs in any different way than current CNS active compounds.
Thank you, David. That was a great overview. And now we'll transition over to the clinical studies that are also become an integral part of the safety evaluation of the psychedelics. So what you've seen are some of the reinforcing and physical dependency studies that are done on the preclinical setting. This continues into the clinical sphere when we're doing human abuse potential studies, which are essentially dedicated studies in recreational drug users that administer single doses of an investigational drug. And we conduct these studies with a placebo control and also in active control. In these types of studies, we are taking a face valid population, so one that's experienced, presumably in this case, with prior psychedelic use or drugs that have psychedelic properties. And we ask a variety of different questions related to the drug taking experience. The main primary endpoint in these studies are an evaluation on a visual analog scale of how much the subject likes the drug, in which they respond either they dislike it or really like it. But there are a number of other measures that will evaluate the subjective drug effects. And as we conduct and we have this discussion, we'll be talking about some of the nuances in these methodological studies. We also do evaluate the physical dependency studies, which oftentimes are included in later phase trials if there is chronic exposure. And we know that some of these psychedelic drugs are being developed for very acute dosing in the clinical setting. So whether or not they will be administered chronically for periods of at least 4 weeks will be determined based on their therapeutic approach. But these scientific and safety questions, needless to say, are important because as we know, with drug dependence and abuse, oftentimes things are taken off-label. So let's dive into the human abuse potential study, and I'll open it up to my colleagues, Denise and Deborah, to talk about some of the considerations that we will be looking at in terms of the positive controls and some of the measurements in these types of studies when we're evaluating abuse potential in the clinical setting.
Thank you, Beatrice, for this question. I think that if we are considering the classic human abuse potential studies, then the first question that has to be answered in the context of abuse liability is, will this particular drug, psychedelic drug, have drug-liking effects in the moment? And then, of course, what we want to do is we want to be able to take a look more comprehensively at the potential for abuse of a novel molecule, in the sense of would the subjects want to take the drug again? Would they have an overall drug liking? Do they experience high effects? What other kind of sensory experiences might they have that would lead them to try to take the drug again recreationally in the context of their outside of the clinical trial life. And so for hallucinogen molecules, I think that we have to consider carefully the type of active comparator. David was mentioning the certainly using an opioid or a benzodiazepine, and I think that that is absolutely the way to think about it. Another question would be, should we consider maybe a second active comparator such as a stimulant? That is a question to be answered. And in terms of the introduction of pharmacodynamic measures in clinical trials, we can take maybe a step back, knowing that this particular field is developing, and maybe we can think about the introduction of some pharmacodynamic measures a little bit earlier in the clinical trials, even at the first in human level, where we can ask some questions regarding the cognitive aspects of these drugs, and whether or not safety should also include a component of cognitive performance. By this, I mean, should we look maybe at attention? Should we look maybe at the ability to react appropriately and fast if we have been given a hallucinogen? And so those are questions that can be answered maybe at the level of a single ascending dose study or a multiple ascending dose study, and that could guide the later clinical trials like the abuse potential trials. And within that abuse potential context, what we probably would want to tap into is the subjective experiences. By this, I mean, how is the drug making me feel? Do I like it? Am I experiencing a high? Am I experiencing good effects? Also, am I experiencing any negative effects? Do I have a bad trip? Do I have a bad feeling? Am I worried? Am I anxious? Those kind of questions could be part of that testing battery, and then combining those questions with more objective measures. Those measures would tap into attention, into maybe even things like facial recognition or learning and memory, and so forth. So those would be the kind of objective questions and measures that one could consider.
Mm-hmm. Yeah, and you bring up an interesting point, Denise, and that's really around drug liking and the kind of psychedelic experience that subjects may undergo when they're in the clinic or when they're taking these types of drugs. These types of mystical experiences can be overwhelming. They can be very positive. They can be spiritual, but they can also be very scary and anxiolytic. And so the nature, when you ask something very general like drug liking, that's a measure where it highly correlates in a positive direction when you have drugs that classically induce the kind of high and euphoria that David had alluded to earlier. But when you have psychedelics, what you're trying to characterize is the degree of these perceptual changes that they induce. And sometimes they may or may not be liked, but nonetheless, even if it's disliked, there's still notable perceptual changes that are related to the characterization of that molecule and the pharmacological effect. So I think to some degree, we really have to think about what are the most important measures when you're dealing with a psychedelic, and is drug liking really precise enough to understand the dynamics of a psychedelic experience? So I think that's one question that we'll be working through human abuse potential testing. The other thing is that because we're testing very high doses, oftentimes we're going into super therapeutic ranges. There's the safety considerations to think about, but there's also the process of unblinding. Subjects who are undergoing high doses and are certainly at the dose levels that will induce psychedelic effects will inevitably be unblinded. Now, we oftentimes that will be the case. They're still under double-blinded conditions, but that's another element that we need to think about in terms of the kinds of considerations, for the methodological considerations. I just wanted to mention also one more point. When we're dealing with some of the second generation psychedelics, if sponsors are modifying existing chemicals such as psilocybin or some of the first generation psychedelics, those may be relevant comparators in these types of studies as they're trying to differentiate potentially signals and safety and human abuse potential with these types of second generation. So those may serve to be very relevant controls in these types of studies. But given the fact that we are dosing to supratherapeutic levels, or levels that we know will be inducing these types of psychedelic properties, we have to carefully consider the medical management of these patients as well. And Dr. Kelsch, I wondered if you might kind of walk us through how the clinic and staff are ready for events that may be sometimes psychotic symptoms or other adverse events that are related to these types of CNS drug effects with psychedelics.
Well, first of all, you have your study populations defined, and I think the more complicated the study, the harder it is to find that particular population. So for example, if we have two comparators, so if we need to use, they have to be able to discriminate between opioids, a benzo, and placebo, you now have a much harder qualification. So not only are they going to have to be experienced in those two compounds, are now also going to have experience using psychedelics and not have had a history of adverse responses. So now you're really limiting your population pool that you start with to those particular individuals. And in many of the studies, it's not that you have to have used it a lot. It may be one lifetime use, and then maybe once in the last 90 days or once in the last year, because some of these compounds are very hard to get. I mean, LSD, PCP, PCP especially easier around here, but psilocybin, I mean, we just don't see people reporting that. So even though they have this one lifetime experience, you're still going to have to give them, as recommended in that compound, a lot of information and sort of what to expect, which is sort of different than what you have to do with opioids and benzos. And so your informed consent process is going to be very important. And surprisingly, like even with, you said the unblinding, you would think everybody could differentiate, let's say, ketamine from placebo, but you still have people who fail. So, so anytime we have a drug that may cause something like a bad trip like this, you would have to make sure your environment is safe, that there's nothing about the furniture or the things on the tables that could be used to harm them. The windows aren't able to be opened, and that you have supervision. I know the recommendation now is one-on-one, but in a facility, a phase 1 facility where we have, video monitoring and just a lot of, maybe other safety officers and paramedics and physicians walking around, can you possibly make it not quite one-on-one and just be looking for the person who actually has the bad trip, because not everyone's going to be getting that compound. So if you have 20 people in-house, they're not all 20 going to get it. So just making sure everyone understands what the compound can do, what are those potential side effects, and to just be able to rapidly increase oversight and supervision for whoever's having a bad trip.
The next question for today is whether or not there are safety and toxicity concerns which are specific to the next generation of psychedelics.
Thank you, Beatrice. Yes, I did allude to the standard preclinical development studies required for an IND for a new psychedelic molecule. And in this question, I'm going to address the toxicological considerations for the testing of new second-generation psychedelic compounds. Historically, there are 2 or 3 major concerns around these types of molecules from historical data and testing. In the development of 2 molecules, dexfenfluramine as an anti-obesity agent, a couple of major concerns came up. The first one was neurotoxicity, which I'm going to address, and the second one was cardiotoxicity. So firstly, just looking at the neurotoxicity and the potential for neurotoxicity of psychedelic drugs, it's important when thinking about neurotoxicity to think about adaptive and reversible changes with compounds in preclinical studies, and irreversible neurotoxic and neuropathic damage. That's the loss of neurons, and in this case we're thinking about serotonergic neurons. And really the molecules in those studies fell into 2 different camps. There are a number of academic groups, so Callahan, Recchiati and McCann, all studied these molecules. And it was generally considered that the fenfluramine and dexfenfluramine type fell into a different category than the MDMA, methamphetamine, MDA type of molecule in terms of their ability to deplete and or damage serotonergic neurons in the CNS and also potentially in the periphery. The MDA type of molecule and the amphetamine type molecules appeared to produce neurotoxic damage with neuropathy and loss of neurons in the forebrain areas to which the RAFA 5-HT neurons project. This was seen by special staining, particularly in primates and mice, interestingly, more so than in the rat model. And this leads us to the point that the choice of the second species in assessing psychedelic molecules is quite important. There was also, as well as markers for neuronal loss and damage, there were also changes in things called the astrogliotic response, a phenomenon called astrogliosis, where the protein GFAP, which I've listed on the slide, actually increases. This is where the astrocytes, the support cells for the neurons in the brain, as well as the microglia, hypertrophy and express this protein, which you can see on the slides on the right-hand side. If you just look at the extreme left one, you can see in the top there's a vehicle-treated animal, and in the lower segment there's an MDMA-treated animal. And you can see that in the case of MDMA, there was a distinct overexpression of this GFAP marker, which is thought to be one of the best markers for neuronal damage in the CNS. There were also other types of changes that were occurring in the CNS. For example, with MDMA, it was postulated and found that free radicals form, particularly the peroxynitrite free radical, which has the potential to damage neurons in the CNS. Some of the studies that were included in these neurotoxicological investigations included the reversibility and the protection against these effects. And some subsequent studies actually showed that, for example, fluoxetine could protect against the reversible changes seen with fenfluramine and dexfenfluramine. But with MDMA, you could only really protect it with, for example, antioxidants, or in some cases, the NMDA receptor antagonists. In terms of the psilocybin LSD class of molecule, there's very little known really about the neurotoxic potential, except that they have the agonist potential and the ability potentially to deplete 5-HT, and thus they do have the potential to possibly cause neurotoxicity in a second generation type molecule. Well, although neurotoxicity didn't lead to the withdrawal of molecules from development, cardiotoxicity did. And as I said earlier, with dexfenfluramine, these were withdrawn from development and from the clinic due to serious valvulopathy liability for psychedelic candidates via 5-HT2B receptor overstimulation. A couple of groups, including Roth and Elangbam, showed that these compounds stimulated 5-HT2B receptors and caused a mitogenesis of the valvular interstitial cells, the VIC cells, in several valve populations of the heart, the aortic and mitral and tricuspid valves. And this led to overgrowth of these cells, valvular insufficiency, and retrograde cardiac blood leakage, leading to pulmonary or aortic hypertension. Some drugs, such as fenfluramine, are still on the market, for example, under the trade name Fintepla, but these have to be monitored. This is used in the treatment of epilepsy, but this has to be monitored quite carefully by echocardiographic monitoring, since it's been shown that a low percentage of individuals do show valvular overgrowth, and therefore clinical monitoring is quite essential. The third type of potential toxicity of these types of drugs is because there are a plethora of cells in different populations of the body which possess 5-HT receptors. They also possess the protein for serotonin transport, which of course is the major site for the target of the earlier generation psychedelic molecules as substrates for this protein. And some of them also contain the enzymes and proteins of the metabolic machinery of the serotonin process. So 3 areas for consideration are indeed the immune system, because many cells of the immune system have 5-HT receptors, and indeed the proteins for serotonin transport and therefore depletion of serotonin. Cells of the how shall I say, the cardiovascular system, the platelets and the endothelial cells, and also mast cells, which have a particular 5-HT receptor subtype, and of course contain histamine and 5-HT, and have the potential to be depleted, perhaps contributing to things like the serotonergic syndrome clinically. The enterochromaffin cells in the gastric mucosa also have large stores of serotonin, and again are targets for these types of drugs potentially in a clinical setting, leading to adverse reactions. And this has to be considered in this type of potential toxicity also. So just to summarize, I think, although we touched upon the, how should we say, the regulatory package needed to get an IND for a new NCE for a psychedelic molecule, taking into account the issues that I've described, that's neurotoxicity, cardiotoxicity, and other toxicities in peripheral parts of the body, I've just listed on this side some of the strategic objectives that we might have to consider in constructing bespoke studies in the early phases of preclinical development. For example, in the early studies, the acute and 1-month tox studies, one has to consider species, because possibly the primate is the most sensitive species, for example, to the MDMA type neurotoxicity. It may also be relevant to the cardiac toxicity in terms of the VIC cells in the valve toxicity that I described. One has to consider new biomarkers to extend into the clinical setting to actually assess the potential safety of neurotoxicity, the immune system, and the cardiovascular system. Moving down the slide, we have to think about lead optimization or plot screens, preclinical lead optimization screens, to fine-tune follow-up lead to selection using investigative toxicological models. And lastly, we have to think about the possibility of designing investigative neurotoxicological models to look at brain chemistry and peripheral chemistry to see if these molecules have the ability to change or deplete serotonin and other monoamines in the CNS, or change the levels of these monoamines and serotonin in the systemic circulation. All these factors from these bespoke early studies could then be used to integrate into the subchronic and chronic tox studies to support the phase 2 and phase 3 development in the preclinical pipeline. Okay, thank you very much for listening.
Thank you, Chris. That's really interesting and certainly very thorough in terms of the amount of investigation that needs to be done from a safety toxicity point of view in the preclinical setting. One idea that came to mind, I was intrigued by some of the antioxidants and some of the neuroprotective effects that can be implemented when you co-administer, and I wonder if that might be an approach for drug developers to co-administer or to combine a drug that may have some degree of neurotoxicity with something like an antioxidant, if there's any kind of mitigation of some of this toxicity, provided that there is a therapeutic benefit to these drugs. So I don't know if that's something that would be reasonable based on some of the data that you provided here.
I think that's a very interesting point, Beatrice. And in fact, I was involved in some of my academic work with trying to combine antioxidant molecules with precursors to dopamine and serotonin, L-dopa, L-tryptophan, for that very reason. And in fact, we never got further into development with these sort of studies or molecules, but there was how should we say, a thought such as the one you've put over, that if you could combine an antioxidant property with a precursor property or a drug property, a pharmacological property, that might lead to protection, as you say, to the presynaptic part of the neuron, which is susceptible to neurotoxicity.
Very interesting. And Chris, are these effects noted upon acute administration, or does this require chronic dosing before you start to see some of these indicators of neurotoxicity and some of the other toxicity that you've seen?
Okay, well, the reports largely from academic work suggest that actually, if you administer, for example, MDMA intravenously, it only requires a couple of doses spread over, I think, about a week to start seeing the neurotoxic or neuropathic potential of the compound. In terms of things like dexfenfluramine, you have to give the compound for a lot longer, for example, a month or so, say orally. And then the early effects are really just, as I say, chronic depletion of 5-HT and possibly other monoamines, which take a long, long time to recover. And there was always a debate about whether there was any associated damage to the neurons, but there were some studies that suggested there was, and there were others that suggested that there wasn't. But certainly with the MDMA type, there was definite damage, as I showed with the GFAP intermediate filament protein, the astrocyte reactions, and also there are some now specialist stains out for looking at neurotoxicity. And I think in the early, certainly in the neuropathological part of or the path part of a 1-month study, one has to consider starting to not only look at brain chemistry, but more specialist stains for nerve damage in the animals, in the 2 species used.
Mm-hmm. Yes, that's very, very good point, Chris. Thank you very much. And I think as we move into the clinical section, a lot of that safety information, a lot of key go, no-go decisions are based on the safety and efficacy profiles in the preclinical phase as to whether or not those molecules will ever make it into the clinical setting. Once they do, it's important to recognize some of this safety information to continue thoroughly investigating it in the clinical setting, whether we may be able to use technologies like neuroimaging, for example. It may be relevant to look at some of the anatomy, brain anatomy, as well as most importantly, a lot of the psychiatric adverse events that I think need to be monitored for some time. So really adequate time to investigate the effects of psychedelic drugs, as well as the aftermath of the psychedelic drug needs to be incorporated into those clinical trial designs. And so as we talk about this further, a little bit about strategy in terms of developing these types of drugs, psychedelics, and particularly the 2nd generation psychedelics. So we see that some of the 1st generations are marked with toxicity, safety concerns. There are probably, these will be very important contrasts and methods to test out in the preclinical setting as you are developing 2nd generation drugs to really determine, is in fact the safety profile improved with the 2nd generations? And we'll leave it to the panel to discuss what would be an ideal 2nd generation drug candidate, and how could we strategically study these types of drugs given the pharmacology of the 1st generations, and what might be some strategies that developers might think about as they're making important go, no-go decisions on their 2nd generation molecule with respect to both safety, abuse potential, as well as potentially even efficacy.
Well, I think if I could possibly kick off the discussion, I think one of the things which is going to be very critical is what level of scheduling is going to actually be applied to the current generation of psychedelic drugs. I mean, we know, for example, that Jack Henningfield and his colleagues wrote an 8-factor analysis evaluating where they thought the risks of psilocybin would place it in terms of scheduling, and came up with the conclusion that the drug should be placed in a relatively low schedule like Schedule 4. We've yet to see whether, if the drug gets approved, whether the FDA and the DEA are going to agree with that assessment, because if the drugs get placed in very restrictive scheduling categories, then there is certainly plenty of scope for developing new drugs which have got much lower levels of scheduling. But on the other hand, if they get placed in a relatively low schedule, then in this particular case, you're going to be looking at drugs which are carrying the same kind of restrictions as maybe, the benzodiazepines, which means the abuse potential is very low, but there are probably going to be very strict limits on the duration of treatment and the duration of intervention that you're going to be able to achieve in patients.
Yes, absolutely. And just for the benefit of the audience, in the US, for example, the drug scheduling process goes from Schedule 1 to Schedule 5, and then there's unscheduled. Scheduled 1 are high abuse potential. They don't have therapeutic benefits, or they have no approved use for medicinal use in the United States. And those will be drugs that will principally be like the psilocybins, the MDMAs, the LSDs. And then Schedule 2 are the same type of high abuse potential compounds, but these are drugs that have approved medical use. So those include drugs like opioids and amphetamines and other classes of drugs that are associated with high abuse potential. As you move from Schedule 3 to 5, they become less and less stronger signals. They become weaker and weaker signals for abuse potential. However, there's still some signal there, and hence they still fall under the Controlled Substances Act. And as you go down from a Schedule, let's say 2 to a 5, the prescribing restrictions are more loose and less conservative, allowing even for the possibility of over-the-counter medications as you get into a Schedule 5, and then certainly unscheduled don't apply to the Controlled Substances Act. So for these reasons, it becomes very important to fully identify and to evaluate the characteristics of a drug as you're collecting data for that NDA filing for ultimately drug scheduling and approval, and to really understand and present it in the 8-factor analysis that David, you mentioned earlier, which is a document and an analysis of that data that the FDA uses in order to make a determination and a recommendation for scheduling, which ultimately the DEA, the Drug Enforcement Agency, will come down to. So I think there is it's an important process, and you're right. I think the primary opportunity is to really thoroughly investigate some of these second-generation psychedelics in relation to the first generation to determine how they differentiate and how they might be different in their abuse potential, and as Chris has mentioned, most importantly, also in the safety profiles. Do they have less of the toxicity that some of the first generation drugs may carry? And then, of course, efficacy will be played out in the later phase trials to see if there may be any advantages on that. So everything is really about risk benefit at the end of the day when we're looking at investigating drugs and determining what their profile looks like, and the data that's collected in well-controlled preclinical and clinical studies then makes its way into the labeling claims for that particular product. And that's very important. So you do need to have well-controlled, well-designed trials, which at the end of the day will be the most important to support the regulatory package and enable appropriate labeling that enables a sponsor or a developer to differentiate their products from other products in the marketplace. So I would like to thank all of our guest panelists today for your time. And I would like to thank the audience for joining us today on this podcast. This will probably launch a series of more podcasts as we get more into this area of exciting research. And we thank you very much for your time today. And thank you for joining. If there are any questions, please do feel free to type these into the chat, or we'll post our email addresses should you have any questions for further follow-up with our team of experts today. Thank you again for joining.